will just say a few things about science and what it is that I've enjoyed in my whole life. When I grew up in South Africa, it was a country that was isolated from the rest of the world. I went to university, there was a war raging in Europe, and there was no communication at all with the rest of the world for a long time. In 1946, it all opened up again. But I had learned one useful thing in life, which is if you can read, you can learn anything. And therefore, I had become what I think is the most useful thing in science, which is not to become a professional, but a talented amateur. So I have been an amateur in many subjects. One subject I got very interested in South Africa was computing. And I had discovered, not so much in Schrödinger as the other people had, but I discovered the writings of John von Neumann. And I still have my copy of the Hickson Symposium, 1951. I should tell you a little bit about that, in which von Neumann describes how you would make a reproducing machine. If you read that, remember this is 2 years before DNA. If you read that, you suddenly realize he discovered DNA, or he discovered the logical mechanism. And later, I did write something on this when I went more deeply into it. But I was very impressed by this. I was also read about DNA. I decided to work in genetics a long time ago. And of course, genetics is connected with chromosomes, so my early work was on chromosomes. And of course, I knew chromosomes contained DNA. And the first evidence, the first place I read this was in the 1898 edition of Wilson's book, the famous book, in which he talks about this nucleic acid being the material basis of heredity. Nobody believed this because they couldn't see how nucleic acids could be so specific. At the time, of course, there was the tetranucleotide hypothesis, and so putting this all together. But I think, at least in my development, the idea of information, that the gene was a message, actually was very much in the thing. And I also had a completely wrong idea of how DNA worked, because there was this big paradox which was noted by almost anybody who was in the field at the time, that the spacing between the amino acids in proteins 3.5 angstrom units, that the spacing between the bases of DNA was 3.3 units. So there was this curious thing that these repeated polymers, and I actually produced a very good system of how you made the protein and the DNA at the same time. by having amino acids linked to nucleotides, just as we have them, and then how this was doubly decomposed. But of course, the problem was, couldn't solve it with that machinery because of the spacing, which is the same thing Gamow found when he tried to do this. But I thought this would be a very good mechanism that you would activate an amino acid with a deoxyribonucleotide and then build the DNA and the protein at the same time. Now, that is, it's only half an idea because it's half right and half wrong. But of course, if it's anything wrong, even if it was 0.1% wrong, it would be wrong. But that's why I call it just a half an idea. I also wanted to work with dyes. I have been fascinated by dyes all my life. I learned how to synthesize them. I made my own dyes. I became very interested in supravital staining. And if you read my early publications, you will see that what I called cell chemistry, these were the things that should have been called molecular biology, but of course didn't exist at the time. So I then finally got a scholarship to go to Oxford, where I wanted to study molecular biology, but of course didn't exist. So I then was directed to a man called Hinshelwood, and Hinshelwood was the center of a great controversy in biology. Hinshelwood was a very famous theoretical chemist, man who spoke 5 languages fluently, including Chinese, and had a smattering of several others, like I have a smattering of English. And he was a very famous scholar, and he wrote a book in 1936, which you should read in the Cold Spring Harbor. It's called The Chemical Kinetics of the Bacterial Cell. In fact, Leslie Orgel, before he died, and I were going to write a paper that said Hinshelwood was the first systems biologist. Of course, you now realize systems biology has disappeared, like many of the other things will in the future. So I then, during this visit, I met Dr. Demeretz. Dr. Demeretz, when he came to talk to me, suddenly realized he was talking to a heretic in the middle of the Catholic Church, in which I believed in mutation. Hinshelwood didn't. And I know in trying to explain this later to Hinshelwood, who accepted it, I then found this wonderful distinction, which I've often quoted, but I'll quote it again to you because it's the most useful distinction that you can make about everything, and that is to distinguish between chastity and impotence. That is the difference between can't and won't. So many of the declarations that people have made to swear that they will never, in all time, change the genetic structure of the human beings, that could be because they won't do it, but it's mostly because they can't do it. so you can make that declaration. I've often, and I've begun to wonder, and I often recommend, don't say never, because I say never, you can never tell what the future holds. There's certain things that you might have to do to save the human race, rather than make these avowals of you don't know that whether you are being chased or just possibly the real answer is you're impotent to do this. Well, I won't take you through much more of this, but I thought last night of something which I think is happening to science, and in particular to our science. When I was in Cambridge, finally, I used to go to give seminars in London. And when people said to me, what are you going to do in London? I said, I'm going to preach to the heathen. That is, I'm going to tell all these biochemists they got the wrong end of the stick. So I then found that that was true. So for many years, we preached to the heathen. Then we found ourselves preaching to the converted, that everybody had become a molecular biologist. And now I'm afraid we're beginning to preach to the preachers of this thing. So it's about time we found new places to go to. But I think the credo, if we can call it that, of molecular biology is first of all the idea, the signal idea, that there's another entity in biology which you don't find in physics. It's information. It's information. And as usual, because you have to handle information in the real world, it's handled in biology by chemistry. So it is, if you like, biology is basically, as I've often described it, very low energy physics, which is what chemistry is. Chemistry is not high energy physics. It's very low energy physics combined with the concept of information. And with that, of course, we human beings have discovered that